Dr. Rick Johnson, and today we're going to talk about Nature Wants Us to Be Fat, Part 3. This is based on my book called Nature Wants Us to Be Fat, and Part 1 talked about the existence of a biological switch that animals use to gain weight and how it's often initiated by eating fructose, which is the sweet carbohydrate that's present in fruit and honey. But in humans, our main source of fructose is from table sugar and from high fructose corn syrup. And we eat a lot of it. And we talked about how this switch is activated in us and is driving obesity and diabetes today. In Part 2, we discussed the surprising finding that it isn't just the fructose that we eat, but it's also the fructose that we make that can drive obesity. And we talked about various foods that can do that. In this Part 3, we're going to talk about how this biological switch may be involved in other diseases besides obesity, diabetes, high blood pressure, heart disease, and that group of diseases. So here we are, Nature Wants Us to Be Fat, Part 3. It's based upon this book, Nature Wants Us to Be Fat, that you can get from many sources. And and I do want to disclose that I have been funded by the National Institute of Health in the USA, and also that our research has led to the discovery and the importance of blocking fructose metabolism as a way to help people. And as such, I am trying to develop inhibitors to block this pathway. Let's hope I succeed. So here we go, part 3. So just again, as a reminder, animals normally regulate their weight very well. If they eat too much one day, they eat too little, they'll eat less the next. But at times, certain times of the year, they may want to gain a lot of fat. And that's often seen in animals preparing for hibernation, for long distance migration, for nesting, and for other situations. And when they do this, they suddenly, it's like activating a switch, and they'll start eating a lot more than they normally do. They seem to be constantly hungry. They're foraging for food. They're reducing their energy when they're at rest. They are putting on fat and weight. The fat is not only in their adipose, but in their liver and in their blood. They become insulin resistant, so that the glucose, instead of going into the muscle, which requires insulin, is available for the brain, which uses less insulin. And this way the animal can preserve the glucose when it thinks that there may not be enough food around, so that the brain gets it first. And a great example of an animal that does this is the fat-tailed dwarf lemur, which lives in Madagascar. And during the dry season, there's not much food around, but even more seriously, there's not a lot of water around, and it will store fat so it can hibernate during those months. And during that time it hibernates, which is actually called estivation if you're in a hot climate, it will actually burn the fat, not just for calories, but because the fat can produce water, as we discussed in Part 2. So what activates the switch for us? What are the main foods that drive this switch, which we believe is the primary cause of obesity and diabetes? Well, the number one, of course, is dietary fructose. When you eat a lot of fructose, you just completely activate this switch. And although natural fruits contain fructose, 1 or 2 fruits, at a time is not sufficient to activate the switch. The intestine can inactivate the first 4 or 5 grams of fructose. And so a natural fruit turns out to be good because of all the good things in it. But if you eat a lot of fruit, a bowl of grapes, or a lot of honey, or sugar and high fructose corn syrup, which is by far the biggest risk factors, then you're going to activate this switch and increase your chances to become fat and diabetic. Well, it's not just that. In Part 2, we talked about foods that can produce fructose, and the big ones were those high glycemic carbs, foods like rice, potatoes, and bread, and how it's accelerated in the presence of salt that salt activates the enzyme called this polyol pathway that converts glucose to fructose. So these high glycemic foods can suddenly be turned into high fructose foods in the setting of a large intake, and especially when given with salt. And then we talked about umami foods, foods that bypass the fructose itself and go right to forming uric acid, and how uric acid can also activate this switch. And umami foods are these kind of high purine, foods that contain like DNA, RNA, nucleic acids, and you can find them in beer where it's mainly in the yeast extract. It can be present in shellfish and organ meats like liver, and we'll talk more about that, I believe. Okay, so here we are. Today we're going to talk about how these foods and this switch have effects that go way beyond obesity. And so let's go back and look at this survival switch, this biological switch activated by fructose. So first it stimulates foraging. It stimulates the foraging of food, hunger and thirst. Secondly, it stimulates fat accumulation. Third, it stimulates insulin resistance, inflammation, blood pressure, and it even reduces oxygen needs. So how does this lead to have a role in other conditions, other diseases? And to look at this one more way, we're just going to look at the biology of this switch, showing how these foods come together to produce fructose, that fructose then gets metabolized. And although it's got a normal calorie pathway where it acts like any calorie, what's unique about fructose is that it activates a special biological switch pathway, and that's driven by the fall in ATP levels that activate a series of steps in which AMP and IMP are broken down to uric acid. The uric acid works on the mitochondria, stimulates vasopressin, and activates kind of an alarm signal by lowering the ATP levels in the cell. So the first one we're going to talk about is how fructose can affect behavior. Now, the foraging response is really when an animal's trying to find food. And it's not just he just walks out there and looks for food. It requires an actual behavior. They have to be relatively urgent because you have to rapidly move through areas to find that food. You can't just kind of dawdle along. You have to be willing to take risks because you're going to go into areas that may be dangerous. So you need a quick reaction time. You need to be a bit impulsive. You can't deliberate on anything very long. You have to be looking around all the time and make rapid decisions. Now, this sounds sort of good, doesn't it? It sounds sort of like what a scout does, what an adventurer does, what an astronaut might have to do. Someone who's, brave and goes into areas, territories previously never discovered or identified or evaluated, and you have to go and you have to be brave and find the food and bring it back to, it sort of sounds like a hero. And so there's a little bit of aspect of this where this is actually sort of a desirable behavior. And you can show this when you give fructose to people. You can use special imaging called bold MRIs and all these kinds of things to look at how fructose affects behavior. And you can show that when you give fructose, it stimulates hunger, it activates the visual cortex, for cues to identify foods. It decreases the activity in the brain associated with self-control, unlike glucose, which stimulates self-control. And when you inhibit self-control, it allows you to be a little bit braver in doing things that may not be so wise, like wandering into a territory that might have a predator, and you're going to be looking for food. So it makes you a little stronger, a little braver when you don't have good self-control. Also, it decreases the blood supply to the areas of the brain involved in recent memory, called like the hippocampus in particular. And by doing so, it can decrease, it can blunt recent memory. And that's actually maybe a good thing if you're foraging, because you don't really want to remember that predator so very well when you're the lion or whatever. You don't want to really remember him vividly if you got to go back. You want to be able to have the desire to still go out there. So it's sort of interesting that fructose says that. Well, what about uric acid? Well, remember that there was a mutation in uric acid metabolism that is a consequence. Humans have higher uric acid levels than most animals. We get a bigger uric acid response to sugar. So when we eat fructose, we make more uric acid, we get higher uric acid levels because we don't break it down as well. And so it's sort of interesting. If you take a mouse and you block that enzyme, so you're kind of recreating what happened to us, you can show that when the uric acid levels go up, the animals become a lot more hyperactive. They become more exploratory. They go longer distances. It's sort of, they become scouts. And likewise, studies in humans show that if you have high uric acid, you're more likely to be excitement seeking and impulsive, and to have whereas there's no stimulation of self-discipline or being deliberating on things. So it sort of fits that uric acid seems to be involved in foraging. Now that's all great, but think about it. If you're activating this process all the time, and you're eating sugar all the time, and you're stimulating these behaviors, suddenly you may not be able to concentrate as well because you're constantly hyperactive, you're constantly like foraging, you're constantly looking for things, you're not spending much attention to detail. And we have a name for this condition. We call it attention deficit hyperactivity, this syndrome or disorder, and it's a condition that is really common. It's increasing dramatically in our population. It's been linked with sugar intake. In fact, many parents think that when they give sugar to their kids, that it stimulates hyperactivity and a foraging response, that it doesn't quench hunger, et cetera, et cetera. So there's a lot of data for it. Actually, I've written papers on this. I encourage you to read them, because I go through the science, and I do think that the evidence is very significant that ADHD is very much linked with chronic sugar intake. Okay. And so again, it looks like it's overactivation of a foraging pathway, a thing that was meant to be good, but when you activate it all the time, it's not. Well, there's another disease that's been linked with sugar intake. It's been linked with uric acid, and it's called bipolar disorder. Bipolar disorder is associated with episodes of mania, sometimes episodes of depression, and especially the manic episodes have been linked with uric acid and with sugar. And so very much like ADHD, it's increasing along as sugar intake increased, so did bipolar disorder. And interestingly, patients with bipolar disorder have elevated fructose levels in their brain. So there's several studies, 2 studies, I believe, that looked at this, and they found that fructose levels are high in the spinal fluid of patients with bipolar disorder, like 2-fold higher than the normal. And this high fructose levels correlate with inpatient relapses, and also these patients often have a high uric acid level, especially during the first attack. So we're seeing some evidence that manic episodes might be linked with this foraging response with hyperactivity and with fructose and uric acid. And in fact, if that's the case, you might think that lowering uric acid might be protective. And there have actually been 4 or 5 randomized trials, and the vast majority of them show a benefit of lowering uric acid on bipolar disorder. This is included when it's usually given as an adjunct to an add-on therapy, but you can show improvements in mania rating scores and remission rates. And so it's something we need to study more. One of the more exciting and tragic associations is the association of fructose and sugar with Alzheimer's disease and dementia. And, Alzheimer's disease is a horrible disease. It affects over 6 million people in the United States. It's, I think, now the 6th most common cause of death. And no one really understands it very well. It's associated with atrophy of the brain and with the accumulation of amyloid plaques and tau protein. And people have been trying to find ways to treat or reduce, to prevent the amyloid plaques or try to reverse some of this. But these kinds of therapies haven't really been that successful. So people are beginning to think, could there be something upstream, something that occurs before the accumulation of tau protein and amyloid plaques? And one of the key findings that keeps coming up is that these people who are going to develop Alzheimer's often show evidence of insulin resistance in the brain. And although we talk about how the brain doesn't really need insulin, that's true for much of the brain, but some regions of the brain are dependent on insulin. So I'm going to present to you this hypothesis that Alzheimer's disease may actually be a fructose disorder. So first off, let's take a look at the risk factors for Alzheimer's include diabetes, obesity, sugary beverages, high salt diet, high glycemic carbs, and processed red meats. All of these are known to activate or to be associated with activation of the biologic switch that I've been talking about. And all of them are expected to stimulate fructose production in the brain. 100%. Okay, so that's kind of scary. Now, when fructose is metabolized, there's an enzyme that is used to make uric acid. That enzyme is called AMP deaminase, and it produces ammonia. And, I'm going to show you some data, but that enzyme's elevated in the brains of Alzheimer's patients, as is ammonia. Okay, and then when you form the uric acid, we already know that it can affect insulin signaling in the muscle. And there's now data showing that this pathway can affect insulin signaling in the brain. And then it reduces mitochondrial function, lowers the ATP levels, causes neuroinflammation. And these are all findings associated with Alzheimer's. And then the idea is that this then chronically leads to neuronal death and injury that can lead to amyloid plaques and tau protein accumulation. So let's look at this fairly straightforward pathway. So the first thing is there evidence that soft drinks increase dementia? And there are studies showing that the more soft drinks you drink, the more likely you're going to have a smaller brain, that the area in the brain that controls recent memory, which is the first place that goes when you're demented, when you start developing dementia, it's one of the first sites where sugar works. And there's even evidence that it affects short and delayed memory. Okay, so we have an association. We also, there's been studies where animals are given fructose. So animals, when they're given fructose, they take a longer time to get through a maze. Okay, that's not good. They show impaired insulin signaling in the brain. Now let's talk about, we talked about how fructose decreases blood flow to the hippocampus. And the hippocampus is recent memory, where recent memory is. And so fructose wants to reduce wants to reduce the blood supply to the hippocampus, which would then impair recent memory, which would help the foraging response. And here we have it. Fructose is blocking insulin signaling. This is one of the few areas of the brain where insulin receptors are really important. And so by blocking glucose uptake into the brain, into the hippocampus, it's going to cause that hippocampal dysfunction. It's going to cause that recent memory problem. And that's associated with oxidative stress in the hippocampus. And that's associated with a fall in ATP levels. Well, that's all scary. That's exactly how the biologic switch works, but it's occurring in the brain And then the ability for the mitochondria to regenerate is also impaired because it blocks a protein called PGC1 alpha. So this is kind of bad news. Fructose actually activates a process targeting recent memory, and it's there to help you forage. But when you're doing it chronically, it's going to cause recent memory loss. Well, this is probably the scariest slide. This slide shows that fructose levels are high in the brains of Alzheimer's patients. This was a study done in which they took 9 patients with Alzheimer's and compared it to a similar number of controls. And what they found was that fructose levels, as well as sorbitol, which is involved in that polyol pathway, and glucose, they were all high in the brains. Now remember that glucose metabolism, the brain isn't burning the glucose as well, so it's expected that the glucose levels would be high because they're not, the brains can't they can't burn the glucose effectively because insulin is being blocked to get the glucose into those areas of the brain that require insulin. So we have high fructose levels. They're like 3 to 5-fold higher, some cases even more, and that's pretty strong evidence that fructose may be involved. There's also evidence that raising uric acid can affect thinking. So again, here is a study where animals had this uricase, this enzyme that metabolizes uric acid. It was blocked. And when you block that enzyme, uric acid levels go up in the blood, but they also go up in the brain, like in the hippocampus on the left. And that's associated with impairment in being able to get through a maze. So usually when an animal goes through a maze, every day it kind of learns a little bit more. So it will get through faster. And you can see in the second figure how the control animals can get through in a faster time when it's on the 4th day, but not if you're hyperuricemic. You seem to have that impairment to remember and to learn. And this is associated with inflammation in the brain, oxidative stress, and also with scarring of that hippocampus, what we call gliosis. Mild scarring can be shown in animals where uric acid is chronically elevated, but also in humans with high uric acid levels. All right. And then I told you that the uric acid is produced by this enzyme called AMP deaminase, and it's a special type of AMP deaminase called AMP deaminase 2, and it is high in patients with Alzheimer's, as is ammonia, which is one of its metabolites. There go up, and it's particularly high in early Alzheimer's when the brain cells are still alive. All right, so I think that based on all the data I've told you, that we need to do more studies, and it looks like fructose metabolism could have a central role in driving Alzheimer's disease, a very frightening finding, and particularly because a lot of these foods we're eating a lot of. And so it really calls for us to understand this pathway better and to determine if this is a major factor driving this terrible disease. Now we're going to move on to alcoholism. And there's a very strong relationship of fructose with alcoholism. And it's been known for decades that people who love alcohol often like sugar, and people who like sugar often like alcohol. The two are very linked. And if you put someone through alcohol treatment and they're in a hospital room and alcohol's been taken away, they almost inevitably want sugar. And so sugar becomes something they can fall back on if there's no alcohol around. So the two have been linked. And Robert Lustig, a great scientist and physician, has argued that, fructose is alcohol without the buzz because you crave it like you can for alcohol. It can cause fatty liver like alcohol can. And so it has a lot of features that are similar to each other. So we talked about the fact in the second lecture that we can make fructose. And we make it through this pathway called the polyol pathway, in which glucose is converted to fructose. And it's converted by an enzyme called aldose reductase. And we talked about how salty foods can activate this pathway. And also we found that sugar can activate this pathway. And we also now have evidence that alcohol also can activate this pathway. So when you have that margarita with the salt on the rim, that's there to help you convert the glucose to fructose, and you've got a little bit of sugar in there, and that's helping, and the alcohol itself is helping. So this is like a triple whammy to make fructose. No wonder we like the margarita. All right, so what's interesting is if you give fructose to animals, you can find the evidence for the enzymes involved in the polyol pathway. They're high in the liver, not just in animals given fructose, but in patients who drink a lot of alcohol. We can find this aldose reductase enzyme. Shown here is this green stain in the livers of patients who are heavy drinkers. And you can measure this alcohol, that in alcoholics you can measure these metabolites like sorbitol, fructose, and uric acid. And if you drink alcohol, as shown in these orange bars, the alcohol-associated liver disease, they're actually, those livers are high in fructose and sorbitol and uric acid. These are the components. So alcohol is making fructose. When you drink alcohol, it's like drinking a soft drink. And it's so depressing to me because I love wine. And, you know, I usually have a glass at night. But now I know that it's actually being used in my body to make some fructose. And, you know, I'm not going to go into it too much, but it turns out that when you block fructose metabolism, you can block the effects of alcohol in the liver. We can block the effects of alcohol that cause liver disease. We can block even the craving of alcohol. And so I'm actively working with the National Institute of Health to try to develop inhibitors of fructose metabolism as a future way to treat alcoholism. All right, so we're going to continue on with that. And we're going to now talk about fructose and cancer. Okay. Could there really be a relationship with cancer? You just told me there's a relationship with alcoholism and dementia and behavioral disorders. It's getting kind of depressing, isn't it? Well, there's also a relationship with cancer. You got to remember that fructose turns out to have a lot of survival properties. And one thing that helps is it helps animals survive under low oxygen conditions. And the reason for that is that when you breathe oxygen, when you breathe air, the oxygen is used to make energy. The oxygen is used heavily by the mitochondria to make ATP. But fructose wants you to make less ATP and wants to keep you kind of in a low energy state. And he wants to put the calories into fat rather than into energy. So the fructose kind of dampens the energy factories, blocks the Krebs cycle and reduces the energy produced. So it reduces the oxygen you need. And that can help an animal if it's in a low oxygen state. And this was shown for a particular animal called the naked mole rat, one of the ugliest creatures in the world, but he lives, you know, in these sandy burrows in Africa where there's very little oxygen. And what this guy does is in these burrows, in order to survive there where most animals can't, it will produce fructose. And the fructose goes up in its blood and in its tissues, and it helps suppress the mitochondrial function so that it can live in a low oxygen state. And so for it's like a wonderful thing to have this because the fructose is helping it survive. But think about cancers. Cancers are these tumors that can spread through the body. And often when they spread to a tissue, initially they don't have a blood supply. So they're kind of in a low oxygen state. So they would like to have a fuel that would help them in a low oxygen state. Guess what that is? That low oxygen state can be helped by feeding those cells fructose. So it turns out that high fructose corn syrup, for example, has been shown to be the perfect growth nutrient for tumors. And in cell culture, high fructose corn syrup really can allow tumor growth very rapidly. And the way it works is that same way. It suppresses the need for oxygen, and so it suppresses those mitochondria, so ATP levels fall, and it stimulates a primitive type of energy production called glycolysis that doesn't require oxygen, and so it kind of switches over to a primitive system, and this allows the tumors to grow in situations where there's not a lot of oxygen. And so when you give high fructose corn syrup to animals, a lot of tumors like intestinal tumors, colon cancer, liver cancer, breast cancer, pancreatic cancer, all these cancers start to grow faster in the setting of high fructose corn syrup. So if you have cancer and you want to do everything you can to slow the growth of that cancer, don't eat a lot of foods like sugar that can stimulate cancer growth. Now, on the other hand, if you're not eating anything at all, it's better to get some nutrients for you, even if the tumor likes it. So if you're really starving, or you have no appetite from the cancer and you're barely getting by, please eat anything you want. But if you are doing fairly well, and you want to keep that cancer down, don't eat a lot of sugar. Don't drink a lot of sugar, because fructose seems to stimulate cancer growth. And that's not just the fructose. We ourselves, we took animals and we inhibited that uricase, that enzyme that mutated in us, so that we raised the uric acid in those mice. And when we did that, we found that tumor metastases were increased. And we could do this multiple ways. So it's also the uric acid that's involved in this process. It's the biologic switch driving cancer growth. And in fact, a high uric acid, as well as high obesity, is associated with various cancers like colon cancer, breast cancer. High sugar diets, being linked with cancers. High uric acid levels are being associated with increased risk for cancer mortality and all-cause mortality. You get the picture? This biologic switch can have a role in tumor growth. It doesn't necessarily cause tumors, but it may stimulate the growth of tumors, and especially the ones that are associated with obesity, like breast cancer, colon cancer, liver cancer, pancreatic cancer. Okay, I wish it would end there, but this biologic switch is also turned on by low oxygen. We just talked about how it's turned on in the naked mole rat. Well, you can have low oxygen in a particular organ and not in another organ. And that happens when you have a heart attack or when you get heart disease. The parts of the heart don't get enough oxygen supply. And guess what happens? The heart starts to make fructose. And again, it probably started off as a way to help it survive. But we now know that it's actually having local effects that are not good. And so for example, in this particular study that was published in Nature, they took mice, they did this thing where they raised its blood pressure and caused the heart to enlarge, and you get this kind of bad cardiac, they call it remodeling, but the heart muscle thickens, it's not good. And when this happens, they could show that this was initially associated with the production of fructose in the muscle, heart muscle, and then it's metabolized and it activates this switch, which is meant to protect it under low oxygen states, but chronically leads to really bad things. And this is some of the data. If you want to look at the data, hypertrophy or enlargement of the heart, they showed that fructose levels go up. The left ventricular mass, or how thick it is, goes up, and you could block it if you could block fructose metabolism. You can't, the relaxation time, it takes a longer time for the ventricle to relax That's improved if you block the fructose metabolism, and you can show evidence of this pathway in human disease. So stay tuned. This biologic switch is having a role in behavioral disorders, in dementia, in cancer, in heart disease, in kidney disease, and probably many other things. So this is a very big switch, and here's the summary. Foods can be high in fructose, or you can make fructose production, you can produce it. And what happens is, shown on the left, normally and under healthy conditions, we get our fructose just from honey and fruits. There's not a lot, there's just enough to kind of protect us. But in excessive situations, like the high sugar diets and Western diet, we're getting an excessive amount. And then what happens is this activates these pathways. This is the biologic survival pathway. Foraging, fat storage, insulin resistance, dampen those mitochondria, increase your blood pressure, stimulate immunity. These are good things, and that's what it was meant for, shown in yellow. It was there to help us search for food, to provide energy and water, to get us fuel for the brain, to decrease our oxygen needs, to maintain our blood pressure, to safeguard us against infection. A wonderful, wonderful thing. But in the world with this Western diet and all these things where we're just hammering ourselves, and with the mutations we've had that make us sensitive to sugar, guess what? This good pathway has turned into a disaster. Now we have behavioral disorders. We have mania, aggression, ADHD, we have obesity, we have diabetes, we're getting Alzheimer's, cancer growth, hypertension, heart failure, stroke, preeclampsia associated with sugar intake, inflammation, atherosclerosis. It is a big story. So in summary, there's an overactivation of the survival pathway. It's going on in our society. It likely has a role not just in obesity and diabetes, but in conditions like alcoholism, ADHD, bipolar disease, Alzheimer's, and as well as cancer, heart disease, and so forth. So in summary, the survival switch has a role in many of the diseases of today. But knowledge of this mechanism allows us to identify ways to prevent and treat it. In my book, I go through everything that we know about how to control this pathway and how to stimulate the recovery of those mitochondria. It isn't as difficult as it seems. We really can do this. A lot of the current approaches like low-carb diet, intermittent fasting, all these kinds of things fit perfectly in with this story. This story explains all the different hypotheses on obesity and kind of explains a lot of disease. And so I encourage you to read the book and to focus not just on what's driving this switch, but how we can turn it off, how we can stay healthy, and how, if we're overweight and have activated this switch so long, how we can reverse it. I believe one day we're going to be able to say that obesity is curable. It won't be a common disorder anymore. Diabetes is going to disappear. And this is all going to happen because of the breakthroughs in science and the advent of ways to break and to reverse this switch. So thank you very much. And this is the end of part 3. And again, I want to thank my collaborators, many collaborators, but Miguel Lanaspa, Carlos Roncal, Gabi Sanchez, Lozada, Peter Andrews, many others. Thank you very much for listening to my talk.